Oh, is it today? Today's okay. Oh, no problem. Can you hear me, guys? No problem, no problem. Sometimes we will experience some internet issues probably because I see there's a east. I mean there's course here. There is some kind of fiber optic breakage recently reported. So there should be some chance that my voice or my picture will be interrupted. It's more on technical side that I cannot do anything. So this is I think yeah, but I will try my best. At least now you can hear me, right? Yeah. In case if it is interrupted due to any reason, please adjust with that. Okay. So is it a holiday? Is it a holiday today then? No, kind of holiday. Okay. Okay. So many of the students, the participant will be attending remotely. Okay, that is good. That is good. Okay. Yeah. Thank you so much. Actually, basically, I will. I don't take much time because I have. I narrow down my slides. I didn't go to, but I will. I will show some basics of. Uh, Probably that it will be also great if the people. You can ask the people to. Uh, close their video streaming because then the band we can it will help us all because otherwise otherwise I mean our streaming will be interrupted all the time. Yes, sir. Are you able to see my screen? Oh, I'm sorry. Let me see. Okay. Okay. Yeah. Thank you so much for a great introduction. Actually, I'm what I'm going to talk more today is more like a mix of it's a blend of a lot of engineering stuff and also a little bit on biology. Actually, each slide need at least like a 2 to 3 hours to explain, in fact, because this is like one of the hot areas in photonics. Photonics means optics and electrical engineering and also biology. Biology means towards the modern medicine. It's a medical application, but it is a topic of research for many of the biology students. There are a lot of students even in my university, the University of California. Several students are still coming and doing even from India and across Asia. Students are coming to join for PhD programs in some of these hot topics because these are very relevant. I believe I made all my presentation in a way that because at least to motivate the people from the different sectors, like there are people who work on physics, okay, especially on optics, okay, they can understand something on it, and also people from some biology and biochemistry. So I made in a way that at least it is beneficial for at least the students and the faculties or from the researchers from the different sectors, different branches, disciplinaries. And thank you so much. And thank you so much for the introduction. And if you have any doubt, because this is a very emerging technology, emerging areas in the field of biomedical imaging, which is a very cross-disciplinary, as I mentioned, it's a mix of blend of many areas. So I will not be going too much detail into certain things. But if you have any doubt, if you want to know more about it, feel free to contact me. You can email me. You can search my name in the Google, and you can find a lot of resources, including how to contact me. So, or you can ask me after my talk. So sometimes you will get only the superficial idea. But if you want to know more about it, feel free to contact me anytime. You can send an email. I will be very much friendly on that. I may not be able to explain everything during the talk, but I will try to answer most of your questions. But feel free to reach out to me anytime, even later, if you are really curious to know more about it. So the outline of my talk is I will talk a little bit more on like different basic medical imaging modalities. So because all the medical imaging modalities based on physics, basically, and how an optical, I'm going to more talk on optical imaging. Optical means we use some light. Using the light, we going to image some biological tissue and how it is useful for the diagnostics of the diseases. That's what I'm going to do. I'll be more specifically talking about optical coherence tomography. So there's an optics coherence tomography. It's a physics term, basically. So that is a imaging technique. It's a very popular. We all use in it. Maybe it is also available in India, our place. People are always used to go to diagnose our eye. If I have a problem with eye, and now it is applicable for different diseases, and it's like really emerging. A lot of several companies in India also coming. It's like in a booming stage now. And I will talk about some optical implementation, and it's little bit. Some of the things will be interesting for you. Then you can take it, and otherwise you can find it something interesting for you. And I will talk about there are several, at least more than 100 applications for this technology. But I will more specifically to the cardiology and ophthalmology. That will be more interesting for you, I believe. And I will give some conclusion about how it's going to be. Okay, some major takeaways about this method. So before going to that, I'm affiliated to, these are my colleagues. I need to show their image because some of the results, so some of the things I will be showing from their research labs. Actually, we have bunch of principal investigators, including myself. I am a part of that. And we have a lot of postdocs, scientists, and other like PhD students from different various countries. And I also want to thank, before starting, I want to thank like many of the research we carried out here is supported by the National Institute of Health, which is a US, and National Eye Institute, because some of my research work is I am focused on eye imaging, imaging eye to understand the diseases of the eye and how eye works. There are a lot of things that I'm exploring in my lab. And I'm housing at the UC Davis. So UC Davis stands for University of California at the Davis. University of California is basically a state university of one of the top R1 research universities in the US. And UC Berkeley, you might have heard how many they have. UC has produced how many Nobel laureate and Nobel Prize winners. Even last year, they have got at least 5 Nobel Prizes in the area, including medicine, chemistry, even some of the electronics. So they are producing we are doing or attempt to do very high quality research in different areas. My area, so I'm more working on like a biomedical imaging. So at least we might be familiar with the different biomedical imaging modalities. When we go to some clinics, we can see a series of different imaging techniques we can use it for. So depend on what we want to image or what the doctor suggested to image. It depends on the disease or the symptoms. The clinicians, they choose different types of imaging modalities. In case of the whole body imaging or the big organ, like they want to scan our head or heart or lung. So such a whole body imaging, typically the clinicians, they use the CT imaging, which basically use of X-rays and MRI scanning and PET. So such imaging techniques are typically used for the whole body imaging or like the organ as a whole. But in certain applications, we don't want to image the whole body, but we want to look only into some tissues. For example, the cancer, detecting the cancer. For biopsy techniques, we don't use the entire image of the body. So we image only some part of the body. For example, some skin cancer. So what the clinicians, they do is like they take a small part or a tissue of the tissue part. They just take some tissues, excise from the, from the skin, and they put under some microscope. So that is the tissue imaging. So typically ultrasound is using for the tissue imaging, and photoacoustic. That is also a very emerging technology in the field now, photoacoustic imaging. So it can go into a few centimeters. It can go, centimeters is like a dream, but it can go up to even like a few millimeters into the tissues and show what are the different structures of the tissue like that. But once we can also look into the tissues even further to see the cells, what are the cellular bodies, or even what is inside the cell and the other granules. So these are very important for the biomedical, for the diagnosis and also for the research application. So OCT is one of such imaging techniques, which I'm going to talk today more about it. It allows for the cellular imaging. So OCT can image the cells, the tissue and its structure and the cells, and also confocal and light microscopy. This confocal and light microscopy is widely used for the biopsy, for the cancer imaging. OCT is these days replacing the confocal and light microscopy to to see the structures of the, structures of the tissues and the cells and how they change. So we can compare the normal tissue, how it looks like, and we can see how a cancerous cancerous tissue looks like. So we can differentiate them, and we can diagnose, oh, this is like a cancerous or non-cancerous, benign. It is called benign tissues like that. And certain application demands like a molecular imaging. That is, we don't want to image the tissues, but we want to see what are the molecules inside it. So the best imaging techniques are Raman imaging. So that's also a very hot topic these days, Raman. So Raman is maybe all of you might have heard about like the Raman effect in the physics. So this makes use of the Raman effect, and using that Raman effect, we can understand how it changes kind of. So Raman, and also electron microscopy. Electron microscopy is like it's almost available in most of the research laboratories. So it really helps to see the structures and also what are the different granules inside or the organelles within the tissues. And if it move further down, that is the smallest, that's like a molecular vibrational imaging. So that is also a very emerging, that is a stimulated SRS imaging. It's widely applicable for many, including the cancer imaging. So the molecular vibrational imaging, which gives a resolution of Armstrong. So we can even image the tissue components which are in the level of Armstrong level. Armstrong means, the 10 to the power minus 10, such a small. So there are, why I show this slide is like, there are a bunch of imaging techniques available in which are typically used in clinics and also in research domains, research labs, to understand from big, that is the whole body imaging, up to the very small, like the molecular level imaging. So there are different types. The most interesting that all these imaging techniques is fundamentally based on physics. So those who are studying physics will be really interesting to explore all this kind of how they work, and also how it can be applied on different tissues, and how can we understand some different tissues. Tissues means biological tissues, and how they change. Using this technique, how can we study different tissues which are like very healthy and non-healthy? That means disease affected. So just make a comparison. I just want to make a comparison on different imaging imaging modality. That's PET, MRI. PET is the positron emission tomography, which make use of nuclear radiation, actually, basically. So this is mostly more commonly used for PET, is used for brain imaging, and some areas it is severely affected by the cancer kind of things. PET imaging is used for MRI and CT scanning. So these are typically used, as I mentioned before the previous slide, it is used for whole body imaging or as the organ as a whole. So it has one of the disadvantages is that it takes long time to acquire the images with this PET, MRI, and CT scan, and it offers very poor resolution. That means 1 to 2 mm for PET. MRI has like 500 to 500 micrometer, that is 0.5 mm to 1 mm, and CT scanning is also similar range. So any tissue structure below this range, that is smaller than this range, cannot be seen by this PET, MRI, and CT scan. This is like a whole body imaging technique. And also, it takes several minutes and hours to get the images. But the good point is that this technique can give some can give the functional imaging. Functional imaging means it can test whether some organ is function or not. And also PET, for example, it can give the molecular level imaging. MRI can also do all this, and CT can also do all this kind of, multiple information like structural. So we can see the structure of our body or the organ, and it also tells whether it is function or not. And it can also give some molecular level information. So these are very important for understanding, how the organ works and what are the changes and how we differentiate with the disease affected organs. So the major issues with, or the major disadvantages with this method is that they offer very poor resolution. On the other hand, ultrasound imaging. Once come to the ultrasound imaging is very commonly used in our clinics. Almost every country they use this ultrasound imaging. So it is for pregnancy time to check the fetal, whether it is growing or not. So we use the ultrasound imaging, and also some people they use it for the thyroid. These days, a lot of cases are reporting for thyroid issues. So many of the thyroid issues will end up with the thyroid cancer. So many clinicians they advise the ultrasound image to see, because it can be easily imaged using ultrasound image. And it offers, one of the reasons is that it offers very good resolution up to 100 to 200 micrometers, which is sufficient to show different structures and different changes in the tissue structures within the thyroid or the fetal or the skin. It's also widely used for the skin imaging. So the OCT on the other hand, this is like a very relatively new technology, OCT, optical coherence tomography. It can offer a resolution up to 1 to 10 micrometer. There are some research now, the OCT image resolution can go even lower, that is lower than 1 micro resolution, sub-micrometer resolution. So it can clearly show different cells in the tissues. So it helps to understand what are the things happening in the tissues, and what are its changes related to the disease affected tissues like that. So this is the thing I will more talk about on the coming slides. And another technique is optical, that is the fluorescent microscopy, which is also commonly used in most of the labs and clinics. So the advantage with the optical microscopy is that it can give a superb resolution. So we can even see very small the nucleus. So you might know, like, there are different tissue components in the tissue components available, like organelles. So every tissue has got the cells. The cell has the nuclei, and the nuclei has subnuclear. So most of the things can be visibly using the optical microscopy, and in addition with the fluorescence. So the tissue usually have some fluorescent property. So optical fluorescent microscopy also allows to image the fluorescent elements in the tissues. So all these are typically used to understand image and understand what are the tissue components in the biological specimen, which is really important for understanding the diseases, whether it is affected by the disease. I'm not talking about the clinical side, okay, by imaging for the disease-related applications. So this can also give like structure. So using this, we can image different structures, and we can check whether the tissues function or not. And also it can explore different molecular components in the tissue. So overall, these techniques provide multiple complementary information about the tissues. So this is basically a comparison about the resolution. So on the y-axis, it shows the resolution. Resolution means how small we can see that. And this is the depth of penetration. So we are imaging the tissue. So we want the light to go inside the tissue and see what is inside the tissue, right? So this is the depth of penetration. And the y-axis, it shows the resolution. So there is a trade-off between this. So if some method gives very high resolution, that means it has shallow penetration. Shallow means it cannot penetrate too much. For example, the confocal microscopy, the microscopy one, it can offer very best resolution. That means we can see even nucleus or even very small elements in the tissues, but the light cannot penetrate too much. It can go only up to 10 micrometers. So if you, at the same time, if you go up to like the photoacoustic tomography, that is also another very emerging technology. A lot of research and it's going on this area in this technique. So it can go up to, it can offer a resolution up to 100 micrometers, but so that is relatively poor than the microscopy, confocal microscopy. So that means it can go up to, it can typically give like a 10 micrometer resolution. That means it has a relatively poor resolution. That means there is an increased depth. That means the photoacoustic can image maximum up to 1 centimeter. So as the image resolution increases, the depth of penetration decreases. So this is a basic like comparison. So the MRI CT ultrasound comes like 10 centimeter. It can go up to even 10 centimeter or even more. Even it can go up to like a meter in some cases. But they offer very poor resolution. So optical coherence tomography basically fills a gap between this technique, very high resolution microscopy, and as well as the higher penetration depth imaging technique. So this is a very optimal position in terms of depth of penetration and resolution. It makes it more applicable for very variety of applications, which is very useful, including skin, eye, cardiology, and I will explain all those applications. So this is very critical, very crucial in terms of depth of penetration and also the resolution. So it can offer relatively good resolution and relatively good depth penetration in tissues. Okay, so this is, I just want to give before going to the OCT, I just want to give a little bit on introduction to ultrasound imaging. So this is a little bit familiar to us. So this is ultrasound imaging. So this is ultrasound probe, and this has put in the eye imaging. Usually we don't use it for the eye imaging. We use for other applications like the thyroid imaging or the fetal for when somebody is getting pregnant. So this is widely used for the such application. But some very rare cases we also use for the eye imaging. So this is the image looks like. See? So when we use the ultrasound, but one point with the ultrasound is that this is not invasive. This is a contact method. This is a non-invasive, but this is a contact method. Contact method means this probe has to touch on the body. So now it is touched on the eye. Now we can see like a different structure. You can see like our eye has cornea. This is a lens, and there is a dark side. That means there is no reflection coming. There is no reflection. That means there is some vitreous humor, some water inside our eye. So that's why it appears as the dark. And there is like a retina comes here, but we cannot see inside the retina because of the poor resolution, because the ultrasound can only image up to 100 micrometer, and it cannot resolve what are the different layers within the retina. Retina comes at the back side of the eye. And how this works basically, I just want to mention this, how this works. Ultrasound is basically works based on a reflection technique, reflection. That is also called echo-based method. So ultrasound makes use of acoustic wave. Acoustic means sound waves, and that sound wave will be propagated into the material or the tissues. And whenever there is some tissue structure comes, it reflects the sound back. So based on the time, propagation, and reflection, it can the receiver can understand that, okay, there is an object at a certain distance away from this transceiver. This is called transceiver because it transmits and receives. So so it's based on the calculation between the distance travel and also the speed of the sound wave used. But one of the problem is that with this technique is that it can measure maximum up to 100 micrometer, because any object within the 100 micrometer need a very, because it cannot detect it, because it need a very high time resolution for it, because it use some kind of electronics. It needs very like a nanosecond range, less than like a 10 nanosecond like temporal resolution. So no electronics can detect at this time. But there are some improvements now, but new technology came to overcome this. But still, with the conventional technique, we cannot image that. So this is the way the ultrasound works, because it's based on reflection, but it offers only poor resolution. But it can show some structures, but it cannot show the structures in very detail. In such cases, the optical coherence tomography is using. So the OCT, this is a general introduction. I just want to. OCT basically allows high resolution cross-sectional imaging of the tissues in 2D as well as in 3D imaging and biological sample in vivo. So these are the certain general characteristics. It is a non-invasive. That means OCT can image without penetrating the probe into our body. So we don't want to really touch or penetrate the probe into our body, but even from keeping some distance away, we can image what is inside our body or the tissue. It can offer like a cellular resolution. That means even any structures in our tissues within the 1 to 10 micrometer can be imaged using this technique. And video rate imaging, it can access the images of the cross-sectional images of the tissues in a very fast rate, like at least like 30 frames per second. Now it is like, even I developed a system here in a couple of years ago. It can go up to like maybe 10,000 frames per second, such as high speed rate. So there are technology improved, like it can access very fast imaging of the tissue because it has a lot of benefits for understanding some tissue mechanisms kind of thing. And also high sensitivity. High sensitivity means it can image even very deep portion of the some of the tissue within the limits. And one of the key points is that OCT can be integrated with other microscopic techniques to explore, to acquire multiple complementary information. There are multiple information also. I don't want to explain all these because this needs a lot of explanation that I will explain later in slides. Okay, then how does the optical coherence tomography work? So the optical coherence tomography is based on the optical interferometry. Many of the physics, those who are studying the physics, especially the optics, they will be very much familiar with the optical interferometry. So optical interferometry means the light mix and they generate some interference pattern. So this is a Michelson interferometer. So Michelson interferometer basically makes use of two mirrors, and we use a monochromatic source. Monochromatic source means a laser source, for example. So when we use a monochromatic, that's a laser source, the light comes and it's split into two-way. One goes to the one mirror and other goes to the mirror two. So from the mirror, these two mirrors, they come back and they combine here. So once two lights combine, that generates some interference pattern. So there is a very important condition here that is, we have to strictly use some monochromatic source. Monochromatic source means it should, that is the laser, it should show some coherence property. So whenever there is a coherence, there will be interference. If there is no coherence, there won't be interference. That's it. So to mix the light and generate some pattern, the light should be like a coherence, or at least it shows some sort of coherence property. So the very important condition for the interference is that the interference fringes generate only if there is the path length difference. So there are two paths. One is light goes here and come back, and here also light goes and comes back. So these are two interfering paths. So the path length difference between these two mirrors, these two paths should be within the coherence length. So this is a very fundamental in physics. So if this condition meets of the coherence length, then how do we calculate the coherence length? Coherence length is calculated, it's based on the source parameter. So every source has a wavelength. This emits the light. So every light has a certain wavelength, and that light source will have some spectral width. That is called number of spectral band within. That's called the bandwidth. So the coherence length is typically calculated as lambda square by like delta lambda. So for a monochromatic source, that is a laser source, this coherence length will be very high. That is like even several meters, because the delta lambda, the spectral width will be very small. So once this gets small, the coherence length will be like very high. That means even if we move the mirror too far, we can still see the pattern because their coherence length is very, very high because it's a laser source. But what happens when we restrict the monochromaticity of the source? That means we use a broadband source instead of a single line. That means this has a central wavelength and a bandwidth. So for example, 25 nanometers. What happens is that the path length allowed in this case will be very much limited, because here the coherence length will be very small. For example, this calculation will be 15 micrometers. That means we cannot move these two mirrors too much far, because if they're too much far, that will be exceeding the coherence length. So we cannot see the interference pattern. So we can move these two mirrors only a little bit away within the 50 micrometer, then we can see the fringes pattern. And if the bandwidth of the source is even higher, we can move only less. That means this property is called the low coherence interferometer. So the low coherence interferometer basically makes use of a broadband light source. That means the interference occurs only from a very small difference in the path length difference. So this technique is typically used in OCT. That is, OCT is based on low coherence interferometry, which allows the light from a very small path difference to interfere, and this property can be utilized to distinguish two different closely packed layers. This is a little bit on the physics. So if you have any, you can also further read, or you can also contact me later in case if you want like more illustration about this one. Give me a second. Okay, thank you. So this shows how a optical coherence tomography works. So this is a Michelson interferometer, and as I mentioned, I use a broadband source which has a central wavelength to lambda 0, and it has certain bandwidth delta. So this is not a monochromatic source. This is a broadband light source. So this is very important for OCT. So any OCT system imaging technique make use of a broadband light source, and it sees a Michelson interferometer. So the Michelson interferometer has this is a translatory mirror. So this is called the reference arm. In this case, we call this as a reference arm. And we place a biological sample here. So every biological sample will have different layers and the clusters of different classes of cells. So it basically makes use of different layers. So how do we image? It's like, so once we place here, X1, and there will be like one position of the layer that exactly matches with the path length, coherence length of the broadband light source, and we see the interferometer. And we move this mirror apart, so we can get the interferometric fringes only from this layer. So the way we move, it's continuously over the longer distance. We get the interferometry signal, very selective interferometry signal only from the deeper portion. This is the way it works. So to get the some interferometry signal from one depth, we have to move the translational mirror. This mirror has to be translated away from the interferometer. So this is shown like a fringes. So what does this fringes means? This fringes basically shows the reflection from here, and this one here, and this one here. And to reconstruct, to get the profile of the reflectivity, we just want to. This is called the filtering. Basically, we use the envelope of this one fringes. So just choose the envelope of this fringes. That basically gives this. It's called air scan. This air scan means the same thing. This is the same thing, but we choose only the envelope or the peak of this interferometric fringes. So this is corresponding to this one, and the second peak corresponding to this layer, and this one like this boundary layer. So this is the way the OCT construct the image. Basically detects the reflection from each layer based on the interferometry. So this is from a single point. Okay, so this is also called point spread function. One reflection is also called point spread function technically, and how small we can measure is depends on the source. Actually, I told you, like if we use the broader, we can separate even very closely packed layers. So it's completely depending on the source parameter, that is like the center wavelength and also how broad it is like. So that is called the bandwidth. So the axial resolution, that is the depth resolution, because we are getting the signal from the depth. So the depth resolution is inversely proportional to the bandwidth of the source. This is more on physics side. Okay. So if you use a source with a much higher bandwidth, we can see even small things because the axial resolution, this will be even very small. That means we can see even fine structures within the tissues. So this is a fiber optic interferometer. This I'm going a little bit up. So this is the way it actually. So you can see here, there's a biological sample here, and this is the fiber optic interferometer. That means here we use like free space. The light is going here, light is exposed to the tissues here and coming back. Everything is on, this is called bulk optics. But this is a fiber optic interferometer. So the light will be going to the fiber optics. And this is most commonly used in the practical scenario. I just want to show this. There is something like this. Okay. So the light is focused. So here you can see like broadband light source. It has a spectral width, delta lambda, and it has a central wavelength, lambda 0, as I mentioned here. And the light comes here, it's split up into two. This is a splitter. So the light goes here. This is like a reference arm, and the other one is going to the sample arm. This is a sample arm, and that light is focused to the sample. So once it's sampled and it is not moving, and we get a fringe like this. See? So once we move here, we get the reflection from the very top, and you can see there are three boundaries, right? Three boundaries, one from air to the tissue, and tissue layer 1 to the layer 2. So that you can see three layers here. So the reflection from, so once we expose the light, when we shoot the light into the tissues, all the light, the light will be reflected from here, here, and here because of the Fresnel, that's called the Fresnel reflection. So because of the refracting index difference, light will be reflected from all these interfaces. So these coming lights will be coupled back to the fiber and coming here, and the light coming from here will be mixed here and generate the interference pattern. That's what you see here. So to get the reflection from different layers, we have to scan the reference arm. So this is a very basic basic demonstration of OCT based on interferometer. This is a fiber optic interferometer. This is the same as this one, but except we use some optical fiber to guide the light and how it's looking. Because in many of the practical applications, we use this kind of configurations. Okay, so I just want to, and so I show this is only from one spot. Okay, so I want to get the similar profile from different region of the tissue, because then only I can construct the image. How do we do that? For that, we use some sort of galvanometer mirror. Physics people also might have studied about the galvanometer. So the galvanometric mirror is set up used for the laser scanning. So here the same thing, we use a scanning mirror. So once the light goes, that light will be scanned across different points across the tissue. That means first it goes to this point. For example, this is our tissue, and first the light is exposed here. We get a profile like this, the same thing I shown here. And next moment, the light will be moved here, and we get the actual profile from here. And using this galvanometer, this is a we call it as a galvano mirror. That means electronically controlled mirror. So using the electronically controlled mirror, we can tilt or we can redirect the path of the light path into different part of the different adjacent points across the tissues. So we get different reflectivity profile from different region, and we convert this reflectivity, because these reflections are coming from different interface. For example, you can see like a lot of different structures here, right? So the light coming from reflected. So these peaks are basically representing different interfaces of the tissue structures. So once we have this profile, we just convert into the logarithm. We have some mathematical operation, logarithmic compression and grayscale conversion. We get image like this. So once you get the OCT image, it looks like this. So this is the actual tissue, and when you image like this, you can see something like this. Of course, it is not a very beautiful image, because you can see a lot of like grainy like structures. It's not really clear because this is a very old image. This is the image created in 95 or something like that. It's a pretty old image, but now we get a very high contrast image because the technology has enhanced a lot. But at least I just want to compare you with this OCT image with this. So you can see the same. This is basically a cancer. This tissue is affected with a cancer. So you can see the cancer here too, right? So these are the image generated using OCT. This is completely based on OCT signal, that is interferometric signal. So this is the way it works. Okay. So this is called a B scan. This is a cross-sectional image. So this is our tissue, and then light falls into it, and we scan the light. We get one OCT image. This is called B scan. And to get a 3D image, we can use two mirrors. This is for one mirror, and if we use two mirrors, we can steer the mirror, or we can drive the light in different B scans. So it looks like this. See, this is from the first position, and this is from the second position. And this is the way we scan the laser across the tissue to get a 3D image. So this is a B scan, that is a 2D image, and this is a 3D image. This is the way it comes. I will show more 3D image in the coming slides. Okay. So OCT is capable of generating images. See, here, very interestingly, you can see we never touch on the tissue. The tissue is here, and we never touch, and the probe is here. We just simply shoot the light on the tissue. The computer, we can see such a beautiful image. And if you scan using two mirrors, you can get a 3D image of the tissue sample, and which is really helpful to understand. Oh, there are some. You can see the surrounding tissue. Okay, that appears differently from here, because this tissue has some problem. What is the problem? Because it's cancer effect, because the tissue property has changed totally. So this OCT image, it is clear that, okay, this tissue has some problem with the surroundings. This is the way it is used for detecting cancer, not only cancer, other diseases as well. Okay, then I talk about the OCT. So OCT has, this is it's not really good image, but I just want to show this is the very first image shown with OCT for the cancer image. That's why I just want to show clearly. This is the 1993 image, first published in MIT in US. So here you can see, okay, now the resolution in OCT. Okay, what is resolution? Resolution means how small we can image, how small things in the tissue we can image. That is called the resolution. So there are two things we can see in the image. There are some axial. Axial means depth in the red color, and there is a resolution on the lateral side. So any imaging system has two types of resolution. One is called depth resolution, and other is called the side resolution, all called the lateral resolution. So in OCT, very interestingly, compared to other techniques, I didn't explain other techniques, but I can generally talk like compared to other techniques. these two resolutions are decoupled. That means we can change this resolution differently from here, this one. But in other techniques like confocal microscope and other microscopes, these two cannot be decoupled. But that means when you increase this axial resolution, that is when you increase the depth resolution, it will affect the lateral resolution. But in OCT, the key point is that we can independently tune the depth resolution. That means we can increase the depth resolution, and also we can increase or decrease the lateral resolution. So they are decoupled. So what are the things they depend? So in depth resolution, basically depends on, as I mentioned before, it's completely depending on the source, the light source we use for the OCT imaging. So it depends on the central wavelength, it's used the bandwidth of the source. And if you choose the appropriate parameter, we can increase the axial resolution or decrease. Nobody wants to decrease, but generally we increase. So these are the parameters we change. So even in my lab and my collaborators' lab, we buy big equipment, big source, which is like several millions of dollars. So that offers very high bandwidth. That means very high bandwidth means the axial resolution delta cell will be very high. That means in the depth, I can see even very small features of the tissues. I can differentiate using, any disease affected tissues, or I can understand more about the tissue, how it looks like. And the lateral resolution is completely depending on the focusing light. In the previous slide, I shown like, okay, I'm focusing here the light. So the lateral resolution is depending on how much I am focusing. If I focus more light, I'm getting very high lateral resolution. This is very important. Some applications we need very high lateral resolution, but some applications we don't really want. We look only the axial resolution. So it depends on the application. So these are very basic equations on microscopy we use. So axial resolution, I told this is the source dependent, and lateral resolution, that is the side resolution. It depends on the objective we use. That means this is the equation. So if we use the wavelength, of course, it depends on the wavelength of the source and all the numerical apertures. Numerical apertures, it's a very important in any objective. You might, if your lab, if in your college you have some microscope, it will be written like some numerical aperture on it. So that means it shows like how much light can be focused. I mean, it is a focusing ability basically. So if it is higher numerical aperture, in the higher numerical aperture, it can focus in a very narrow, small spot. So if this spot is small, that means we can resolve such a very small spot. That means we get a very high resolution. That's what. So for a high resolution, we need a very small spot. Okay. So it depends on the application. Many applications we don't want very high lateral resolution, but we need very high axial resolution like that. So this is the way it works for OCT. Okay. So the one I work, the one I previously shown is like, this is a fiber optic. We have a fiber coupler, light source coming, light split into reference arm and sample arm. So this type of OCT, the OCT, the interferometry I explained is called time domain OCT, because here to get the depth signal, we scan the mirrors over the time. So once we move the mirror over the time, we get the depth of the sample over the time like that. So this is a very old type of technology. Nobody used these days. And this is the first image of the retina actually. Using this technique, in 1991, the Fujimoto, I think most probably within next 5 years, he is going to get the Nobel Prize in Physics for this one. So we are waiting to get this Nobel Prize for this invention. This method is invented in MIT, Massachusetts Institute of Technology in electrical engineering, but they all are physics. It's nothing to do with electrical. They all are physics guys. And they developed this system, interferometry, and they use for imaging. This is the first retina image using this technique in 1991 in MIT, the Fujimoto. So it is a David Holm. He works in Oregon State now as a Fujimoto. He's a little bit old now. And this is the first image. So the image is 99. That's why the image doesn't look like, so you can see a lot of structures here. I mean, there appears in red, some are like green. It's not really beautiful, but at least you can see there are some tissue structures. But why they are colors? Because the color based on the reflection. Some tissue structure reflect the light back very high. That's why it appears as red. Some they don't really reflect in that appears like green. So these are like a color coding. This is just for a representation. I just want to show this is the very first image. So this is the time domain OCT. Now nobody uses this because we need to scan the mirror. That basically reduce the speed of image acquisition. So it will be very super slow imaging technique. So nobody wants to use these days. So on the other hand, there is a new type of we use the same technique, interferometry, but there is something called Fourier-domain OCT system. So what is a Fourier-domain OCT system? I can directly go into that. So in Fourier-domain OCT system, there are two types of system, that is spectrometer-based and set source-based. In Fourier-domain system, we don't use the scanning. We don't scan the mirror. So we have the interferometer, we have the broadband light source. That is must. We should not change it. We need an interferometer. We split the light into reference arm and sample arm. And the light coming from the mirror come back and the sample come back, then interference. They produce some interference, as I shown before. And previously we detect using some photo detector, and we just extract the envelope of each reflection to get the F10 profile, that means the depth reflectivity profile. But in this case, we never move the mirrors. We keep the mirror static. So then what happens? So we have a sample, we have a mirror, we never move it, and we still get some interference pattern. But in this case, we construct the image by spectrally resolving the signal. This is a little bit deep into the physics. That's like at least take 1 or 2 hours to explain. But I can just go into the brief. So in this case, we spectrally resolve. We don't want to move the things, but still we get the depth information of the sample. Depth information means different tissue layers of the sample by spectrally resolving the components of the broadband light source, the interferometric signal or the interferogram. And we further process. How do we process? We perform a Fourier transform of this. So just by performing the Fourier transform of the interference fringes, we can still reconstruct the image. So this technique is called spectral interferometry. This is a different topic in the physics, basically optics. It's very interesting, but we can still get different things. But the only difference is that here the depth is recognized or differentiated as the frequency of signal. So here there are multiple, I can give a little bit more on it, because there are multiple frequency components, because it has a multiple wavelengths. All these multiple wavelengths generate interferometric pattern from different depths at different frequencies. And at here we get a composite signal, but in the composite signal, we can split up different frequencies using Fourier transform. So just apply the Fourier transform, we get the peak from different layers. So for example, this sample has 3 different reflectors. For example, it has 3 different layers. So the first layer comes as a low frequency. If you go deeper, the reflection will come as a higher frequency. So once we detect here, we get this like a single signal. But if you just find the Fourier transform, you get the 3 different peaks. So these 3 different peaks represent, one peak is from here, the first place in reflectivity, and second from here, and third from here. But you may think that, okay, this has higher peak, but this has less peak. But it depends on the reflectivity of the, some structures are reflecting very high. That means we get high peak. This is less reflecting, that means we get lower. But this again, this reflects better than this one. That's why we are getting very high peak. It has nothing to do with the frequency, but it purely depends on the reflection. So in this method, basically, still we use the interferometry, but we don't move the mirrors to get the the coherence length matching or something like that. Instead, we use the in spectral interferometry, where we perform the Fourier transform of the interferometry to get the depth information signal. This is a very important technique, because it's a very latest technology. In almost all the new technology, either use of this one or this is a fiber optic implementation of this one. I don't want to explain this one. I can just pass all this one. And the other technique is the set source based OCT system, where instead of using a broadband light source, I'm using a tunable narrowband light source. That means instead of using the entire spectrum, I will use a sweeping source. Sweeping source means it will sweep the wavelength from one band to one wavelength position to the other band. Eventually, the wavelength will be sweeping across the bandwidth. So still it works as a broadband source. Instead of we illuminate the whole system using the entire broadband source, here we use a scanning wavelength, basically. So we tune the wavelength across the bandwidth and collect the information. So this is called set source OCT system. You can also find some articles, and this is very basic. This also uses the spectral interferometry. Here also what we do is we collect the interferometry signal from different depths, and we just do the Fourier transform as I shown previous slide. The same technique. Instead of using broadband source, we use a tunable light source across the bandwidth. That's it. There are more things to speak about here, but it may take longer, so I'm just keeping to give only a brief idea about it. Okay. Then that means the OCT can give a cross-sectional image and also 3D image of the biological tissue. So it can go up to from 1 micron to 10 micron meter, but it has a penetration maximum up to 2 mm. That depends on the tissue. Some cases it can go up to 3 mm, but most cases in skin or some thick, optically thick tissues, it goes maximum up to only 1 millimeter only. So these are some of the major notable applications. There are tons of applications, but I'm just mentioning only notable applications of the OCT where OCT is applying. The most prominent application of the OCT is the ophthalmology. So this is like a very prominent and most dominant application of the OCT. So if you have any eye problem, if you go to the clinicians, they do some preliminary tests with other techniques. That's very simple. They don't charge you much. If they found something serious, they will ask you to take the OCT image. So almost every clinician has the OCT, but OCT is still developing. It is still progressing in very high. Maybe the clinical using still use some old techniques, but it has revolutionized the ophthalmology. Even any eye diseases now we can detect even very small earlier stages. So ophthalmology is one of the prominent application of the OCT. That means the eye can be imaged. That I will explain later more. Then dermatology. What is dermatology? Skin imaging. So OCT can also image in vivo. Like in vivo means without removing the tissue. We can tissue within the body. We can directly image. So this is example. So this is the tissue. So when you use the OCT, so the image looks like this. See? So you don't want to touch the OCT light on the body. You can just keep like a few centimeters away. Just shine the light into the tissue. You can see the image like this. You see, this is still not a great image. You still have a very good images. You can see on the internet. If you browse in the Google or something, you can still see some skin images taken with the OCT. So you can see one structures here. These are taken with the OCT by scanning the OCT beam across this tissue. So this also helps because if there is some kind of abnormality, malignancy in the tissue, malignancy means something like a cancer. You can see this is a very healthy tissue. But if there is some kind of malignancy, you can see the structure will be totally different. So that means when you scan it with the OCT, you can see that, oh, there is some abnormality, something different appears there. That means there is something growing there. So this is a way diagnosed for the skin. So this is widely used for dermatology. In our Kerala also, Amrita University, they have widely using this OCT for the dermatology applications. For the screening skin cancer approaches. Yeah, they recently published one paper with that clinical study. And this is another important application, cardiology. Cardiology means in India especially, like a lot of people are dying with the myocardiac arrest. That means heart attacks. So this is basically caused by the deposition of the cholesterol. That's called the plaque. It's technically called the plaque within the artery wall of the artery wall, the coronary artery walls. So there are different angiograms. We typically use the angiogram and angiography. So now the OCT is replacing. Previously, the ultrasound was used for imaging the coronary wall. Now, also in ultrasound, all the ultrasound, intravascular ultrasound is completely replaced by the OCT, intravascular OCT imaging technique. I will show a couple of images. Then it's also widely used for the dentistry. So this is a healthy teeth. You can see like, okay, very healthy. It looks like normal. But here, there is some cavity. So if there is a cavity, if you put the OCT, you can see the cavities appear here. So all the defects within the biological tissue can be easily observed within the B scan. This is called cross-sectional or B scan from the OCT. So this is also widely used in the dentistry these days. And endoscopic applications. Endoscopy now is emerged as like to detect colon cancer widely used for, because now the OCT, I show you there are two arms. One is reference arm and the sample arm. So the sample arm can be put into a catheter, and the catheter means a tube connected with the light optical fiber. That's called a catheter. So the catheter can be put inside any hollow entry. That means like a colon or any invasive part of the body. So they can put into the medical cavity. Then it can go inside and image. So this is a very useful imaging technique these days in the colon cancer. You can see there are some images. I didn't show like in the bigger picture. You can see these are very healthy, but you can see in the cross-sectional there are some lines here. That lines is basically the cancer, colon cancer coming from this. So this is also imaging widely used for the colon cancer these days as an endoscopic probe. And now it's also for the embryology. Embryology means like the developmental stage, the baby is coming, like the fetals. It's also widely applicable for the fetals, imaging fetal these days, for the small animal, not for the human, but for the some research application like zebra fish. There are a lot of people working on different animals to understand, how the biology mechanism works. So they use different, including mice, rats, and also like zebra fish. So in such a small animals, OCT is also used for the embryos, to study the embryos. And there are some color techniques that shows that its heart is functioning. So it can tell a lot of function, because OCT also allows the functional. Not only display, but it's also tell whether it's functional or not. Yeah, I mean, I just want to give you like it's widely applied for different applications. So in future, some of you at least can want to do some research in PhD or something like that. You can choose any of this, and you can also use OCT, some images, and understand something like this. So you can choose some of these areas for your future studies. Okay, so these are the increasing applicability of the OCT over the years. I showed only up to 2018, but there are up to 20, even now 2025 available. This is a little bit older slide I used, but you can even from this slide you can see there's a clear trend coming. So there are different colors and years. Years means okay, now it's over the years how it changes. The colors basically different applications. So the ophthalmology still remains as the most prominent application, but the applicability of OCT also extends to other areas. For example, the blue is show the cardiovascular. Now the cardiovascular OCT is the standard technique, and also like dermatology. I showed like a couple of gastroenterology, neurology. See, you can see all bunch of different applications where the OCT is applied. So this is very promising for biomedical applications. Basically, OCT, that's the advantage. OCT purely based on physics, light, optics. So how it is applied on detecting diseases for different biomedical applications. That's very, very interesting. And okay, I would like to start with how it is applied to the, I want to explain a couple of applications. I will be explaining two. One is the interventional cardiology, how it is applied on cardiology, and also ophthalmology. Ophthalmology, I take more time because cardiology, I will just glance through it, but ophthalmology, I will spend more time on it. So this is a coronary artery, how the OCT is applied for the coronary artery imaging. So this is basically a cartoon or the schematic of the coronary artery showing the anatomy. In our heart, there are the blood vessels. The coronary artery basically supply the blood, oxygenated blood to the muscles of the heart, right? So how this cardiac arrest happens? Because if there is any block in the coronary artery, it blocks the blood that carries oxygen. That means the tissues in the heart, they lack the oxygen and they cannot function, and it end up with cardiac arrest. That's the way it happens. So any deposition, so how it blocks? Because in practical cases, because our food style, lifestyle, we eat a lot of cholesterol-rich foods. What happens is that the coronary artery is the place where it is more deposited. So especially at the junctions. So these are the coronary artery, and they split up into different branches, and the cholesterol will deposit all the major walls. Then what the problem is that then it starts accumulating, and then it reaches a certain point that it blocks the entire blood flow. That means the tissues will never get any kind of oxygen to blood, and then it end up with breakage of the tissues. That means end up with a heart attack. That is called simply the heart attack. So typically in our place, actually in the clinics, basically we use the coronary angiography. That is X-ray based. Most cases we use for example, somebody experiencing the heart chest pain, and he will be taken to the hospital. First they will do is like an angiography. So how do they do that? So they use simply a CT scan. There will be like X-ray or CT scan with that. So they inject a special dye into our blood, and then that blood will be spread everywhere, and we image only the coronary artery. So the imaging will be focused on the heart. So you can see that this is like all the blood vessels. This is the coronary artery, but at some places you can see it doesn't show much. That means the blood is not flowing there. That's why it is not showing that. So this blood is basically carrying the dye. So we will inject something called a dye. Dye means like contrast agent. So we can see this blood vessel only if you inject that. In the hospital they call some kind of medicine. It's not really medicine. It is called the contrast agent, or it is called the dye. So now we can see because the blood is carrying the dye, but we can see here there is some problem. We cannot clearly see it because there is some block. So this is the way the doctors detail, there is some block here. Then what they do is like they do some kind of angioplasty. Angioplasty means they put some stent inside it, and or it's also called balloon surgery. There are different ways to do it, but the most advanced method is like metallic stent into it. Then once the stent is deployed, that means it enlarges to the normal level, that the blood starting flow. So now you can see after the stent implant, you can see this is like a black color. Why? Because the blood is start flowing here. So this is a coronary artery I just shown. This is not OCT image. This is like a X-ray image, normal image, other imaging technique, standard imaging technique we use. Now, how it is detected. So what is the role of OCT in coronary artery imaging? So this is the most important because the doctors really want to place, once they detect, this is called the screening. So once the patient is taken into the hospital, so what they do is like they inject the dye and they image using the CT scan, and they can find the blocks like this. There are a few places they can see the blocks. There will be some cases 6 blocks, 8 blocks, 10 blocks like that. They can see that. Then, so this is called screening. Then what they do, they want to look these places. So how do they look? The most common places in even in India or other places, like what they do is this is called intravascular ultrasound imaging. So most of the hospitals these days has the intravascular ultrasound imaging. So what they do is they put a catheter, they put a tube inside it, and they will pass that tube through the blood until it reaches to the heart vessel. That means the coronary artery. So once it is moving through this blocks, you can see. So you can see something here. So the surrounding part is the blood vessel. It is the circumference. It is the circumference of the blood vessel. And at the center, you can see some dark spot. Dark spot means it is the hole inside. And there is some white spot here. That is the reflection because it is a catheter. It's the tube. I will show you in the next slide how it looks like. So when it moves, there will be some signal coming back. Okay, so this is nothing to do with it. This is a probe. This is the reflection from the probe. So when it moves, you can see there are something here. So somewhat like deposit, the cholesterol deposit, but it is not clearly seen. But if you use the OCT-based intracoronary imaging, you can see the deposit much more clear here. Okay, so again, you see like some round here. That is this is the blood vessel wall, the wall of the blood vessel, and this super shining area is the cholesterol deposit, and you see something like this. There is a dark. This is a catheter. Okay, there's a tube. What is it called? There is a metallic stuff to support the tube into it. So this is the one, that one, and this is some back reflection from the probe actually. So this has nothing to do with the blood vessel. This is from our optical components. So this clearly image, now we can see in a better image than this one. So because the OCT offers high resolution imaging. Okay, I just want to compare this one. And this is another sort of images. So this also shows there are some, see, if it is a good, there is no block, it will be exactly looks like circular. But you can see this is not circular because there are some cholesterol deposit across here. So, and we can see with a very high resolution. And what the doctors do is like when they appear across, so they put the catheter and they move inside the blood vessels. And when they move, it will start imaging on the walls. That's the way. And they do is like they stretch the image in a linear way. So now you can see the blood, how they deposit. So this is the image comparison between intravascular ultrasound and this is called the OCT imaging, the optical imaging technique. So you can see the difference. So in the ultrasound, you can see, okay, there are some blocks, but you cannot see much clearly because the resolution is so poor. It can go up to maximum 100 micrometer. But in the OCT, you can see like, okay, even the deposit is much more better clearly visible. And at the same time, it can even clearly each and everything. So the OCT basically helps. So why they taking this one? Why they take these images? Just to understand how much deposit is there. And also this helps to put the stent. Because when they employ the stent inside the blood vessel, they want to really look at what is the size required, what is the length required. So basically, this OCT-based intravascular imaging helps to measure or decide for the doctors to, what should be the size of the stent to be deployed in that. So this is a really helpful. So that's why it's called interventional cardiology. Okay, so this is how they do. Okay, so this is the blood vessel of coronary artery. This is our OCT. So as I showed, like this is a reference beam, and we use laser, not laser. We had to use a broadband light source, and this is a reference arm. I just purposely use the laser. Okay, so the the sample arm light goes here, and we put it a catheter and we pump into the blood. So it goes here all the way, and you can see there are some blocks, and we still pass through it. On the way, it image like that. So how? So this is the prop actually. So these are the typical prop we use for intracoronary imaging. So this prop will be put into, this is a very small, this size is very small, that will be fit inside a coronary artery. And during the imaging, we pump this, we push this prop inside the blood vessel, and we'll keep pushing until it reaches the block. Okay, so you can see here, there is an, these are like some mechanical structures, optics, there are a lot of optics, a lot of lenses inside, but you can see like there is a beam here, light beam here. So when it moves through the blood vessel or the coronary artery, this will circulate, this will rotate. So during the rotation, it captures this image. So how this captured? Because when it moves, it is called circumferential scanning. So it performs the circumferential scanning to get the image of the walls. That's the way it appears. So basically, I just want to show this. This is the way it looks like in the OCT imaging. So now in most of the hospitals, they charge a lot. Now they ask, if somebody is taken to the hospital, they charge, you want ultrasound imaging, poor quality image, or you want high resolution imaging with OCT for the country. So if they take with the OCT, now they say from a hospital, they charge very high because it offers, it's a sophisticated technology, and it offers very, very good quality image. That's the reason. And this also basically compares. So this is an OCT image. There's a huge, I think this is taken from a patient. First two are taken from two patients. Both of them died because of the cardiac arrest. So when they image, this is the OCT image of the cholesterol deposit in the coronary artery. See? And this is the histology. Histology means after the patient has died, they took the coronary artery and they cut and compare with the in the image to characterize. So here you can see this is the physical, his coronary artery site, and only this much size is open, and all these are cholesterol deposits. So that can be clearly seen here using the OCT, clearly seen. And this is another person. This person is the B. The B. This is a heart block measured from a person who is 74-year-old. So there is another problem here. Once the person is getting old, there will be, of course, there will be some kind of deposition of plaque. That means the cholesterol. In addition to that, there will be calcification. You can see some additional structures in this case that we cannot see here. This is from a 48-year-old guy, and this is from a 74-year-old guy. But once the people get old, there are some additional structures we can see. These structures cannot be seen with ultrasound image. This can see only with OCT image, optical coherence tomography image. So what is this image? Where does this come from? These are called the calcification. These are the calcification, calcium deposits. So in blood vessels, there will be not only the cholesterol, but there will be also calcification, calcium also deposits. That's why the doctors also strongly recommend for the heart patient not to take too much calcium product like milk or any other things, because once they get age, not only the cholesterol deposit, in addition to that, there will be calcification, calcium deposit also. But the problem with the calcium is that even stent cannot suppress it. That's the problem, because the cholesterol can be like a very jelly. Cholesterol is typically like a jelly. So if you put some kind of stent, metallic stent, it will move away. It will move close to the wall, and it can open the coronary artery to make the smooth flow of the blood vessel. But if the calcification is that the calcium is like a rock. Rock means it's like a very strong rock. So even if the stent is deployed, it will not work. So doctors recommend we cannot put the stent because you are too old and there will be calcium. So that's very important. So for the doctors really want to see there should be calcium or not, it cannot be seen with ultrasound conventional imaging technique. It can only see with OCT imaging. That's why some patients are escaped when they go to some top hospital because they have this facility. But the people, even sometimes the patients, they taken into some low profile hospital, they have only the ultrasound image. They will do the same thing, but they cannot see the calcification because it's not the problem with the doctors, because the problem with the imaging system, because the ultrasound cannot see this because it's a poor resolution. So once they use alternate technique like bypass surgery or something else go for that. But the main point I want to emphasize is that using OCT, we can see this kind of different structures within this. And this is the cross-sectional after the person has died because of the cardiac arrest after a few years. And that person is a donor. So he allowed to take out this part, donated the coronary artery, and they just cutting and they compare with the OCT. See, this is the calcification. So we can see exactly what are the changes in the tissues exactly as it is in the OCT image. That's a very, this is from a healthy person. See, you can see the healthy person. It is like there is a slight deposit that it will be common for every person. There will be slight deposit, you can see, but it is almost open. So this is for a healthy person. So now you can see like how a OCT imaging can differentiate a healthy person with the coronary artery images of a healthy person with some age, different age, like old people, and with severe cholesterol deposits. So yeah, this is just to show that. Okay, that's about coronary artery, how it's works, and I just want to show a couple of images for the OCT in ophthalmology. This is a big area. This is people are doing a lot of work. A lot of companies are there now in working. A lot of research groups are there in the top. We are one of the probably the top in the field, not only in the US, in the world actually. We work on eye imaging. So we use a lot of optics. We build the new instruments. We use a lot of physics, engineering, electrical. We use a lot of signal processing. We use a lot of math. So we build a new system to understand eye and eye diseases at earlier stages and how a eye functions. So there are a lot of things we can do with this OCT imaging. So OCT has a broad application in ophthalmology. So this is the representative eye, actually. This is the image of eye. The left side shows the image of the eye, cross-sectional image of the eye. This is how it looks like. So eye has a two part. One is called anterior chamber. Anterior chamber means this is the way eye looks. So this is the cornea, and there will be like some liquid is called aqueous humor, and this is the lens. So when the light comes, this lens will focus the light into this region. It is called the fovea. So the light will not focus anywhere else. Light will focus only this place. So when it focuses here, we can see the object. That's the way it works. So typically the anatomy of the eye is like it has two parts. One is the anterior chamber. This is the top part. We can see. Okay, this is the cornea and lens. Up to this is called anterior chamber, and the back side of the eye it is called the posterior chamber. So OCTs image both anterior and posterior because there is a slight difference in the imaging. Because for the anterior imaging, we typically use 1300 nanometer near infrared imaging because that light cannot go to the bottom because it will be strongly absorbed by the water because there is a lot of water inside our eye. So that will strongly absorb. So the 1300 nanometer is typically used for anterior chamber imaging. So this is how it looks like. Now you can compare. See, so we have the cornea, we have some water inside. This is called aqueous humor. We can call it as water. Okay, and there is a lens here. So how it looks like in OCT, this is the way it looks like in OCT. Okay, so you can see again the cornea, and there is some structures called the iris. Iris is basically control the pupil size. So if there is too much light, we will shrink our eye, right? Because the iris will automatically help us to shrink the eye, make our eye smaller. Okay, so this is the iris. It can be clearly seen in the OCT image, and this is the lens. So it will be look a little bit bigger. Okay, so it has nothing to do because the OCT has a different scale, image scale. So the image scaling will be different, but you can still see the crystalline lens, the lens in our eye, and all these things. Like, okay, so this is a typical OCT image. So doctors, when you go, if you say that, okay, I have some problem in the cornea, I have some irritation, or I have some problem intraocular pressure or some issues, they immediately ask you to take some images with the OCT. So that OCT image looks like this. If this is a healthy eye, so if some person is very healthy, it looks like this. Wow, this is good. Everything is perfect because the iris is here, lens is here, everything is intact. So this is good. But if somebody has any problem with the eye, they can immediately see from here, okay, there are some issues here. And this is the way it looks like. And why this is very important? Because there are some, especially in Kerala, actually, we have a lot of problems with the glaucoma. The people, even it is typically comes at the mid-age, but now it seems like it is reporting. I read some report from the health department of Kerala government. Like they have some statistics shows that even for the kids, now there's an increased number of glaucoma from Kerala because now these days we use a lot of mobile phones all the time, mobile phones, laptop. We watch a lot of movies. We spend a lot of screen time. So because of that, probably one of the reasons, I don't know exactly, but nobody knows, but the health researchers are still investigating why it causes even for the smaller age. Because the glaucoma is basically caused by the intraocular pressure. So you can see here there's some water here that is called aqueous humor. This water will be circulating through so the water will be here, and this water will be circulating through the posterior chamber, and it keeps circulating. That's the way the eye works actually. So there's a water always circulating within the eye, but in glaucoma, there are different types of glaucoma, but the most commonly glaucoma is the closed glaucoma. It happens because there is a corneal angle. There is this water flow will be blocked by some effect here. So due to some changes in the eye here, this is called the iridocorneal angle. So something happens here. The water flow will be blocked. So what happens? This water flow will, the water cannot flow and it will start accumulating and it huge pressure on the eye. That's why some people feel that I have some sort of like some pressure on my eye. Some people, if you spend too much time on the computer or phone, you will have some kind of pressure. That is the symptom. That's the beginning of the thing. Okay, it may not be because of that, but it can be. It can be in future or later. It can be to the glaucoma, but that will experience because of the intraocular pressure. Why intraocular pressure? Because of this blockage of water canal. So to understand this one and to give some treatment, the clinicians, the ophthalmologists need to image here this side. So that's why they use the anterior chamber imaging. So they recommend for the OCT imaging, anterior chamber imaging, and using that they can clearly see some changes here. And using this one, they can make some holes here. That is called keyhole surgery. I mean, that's not typical keyhole surgery. This is related to the eye one. So they shoot some laser to make some holes here and make the water circular as normal like that. So this is very much helpful for doctors to detect the glaucoma and also for their treatment. And also this is used for the cataract surgery. What is cataract surgery? Cataract means the lens, replacing cataract surgery is after 40 or 50 years, most of the people have to do some kind of cataract. So cataract means they replace the lens with the artificial lens. So the lens will lose the ability to focus properly, and it will also become like a haze. Haze means cloudy. So we cannot see anything properly. So once people become aged, so it will be most common during this. So OCT also used to analyze or confirm there is a cataract, and also it's used for during the surgical operation. Also, it is using. So this has a lot of application. Now the most important part and is like a posterior imaging. That is the retinal imaging. This is the most crucial part in the OCT. So we have the retina. Retina basically comes as the posterior part of the eye, which has the back side. Without retina, we cannot see anything. So retina is the most crucial part. Everything is crucial for a good vision. For important to sustain a good vision, we need everything to be perfect. But without retina, it's completely dark. It is completely blind. So if all other parts, for example, lens, cornea, everything can be replaced, but the retina cannot be replaced because it is not yet invented how to replace it. There are some researchers, our collaborators, I mean, my co-researchers are working on stem cell therapies. If there are some photocells destroyed, we have some techniques now to replace them. So we are testing it now. So there are some new emerging technologies all over the world, especially we are also very much working on it on small animals like mice, and also some are we are clinical trials on humans. But the most important part is that if there is no retina is not functioning, retina has some problem, vision has lost forever. We cannot retrieve the vision. No vision means we lose the beauty of our life. That's the thing. So this is how a OCT image looks like. So typically for all the OCT imaging of the retina, we use 850 nanometer or 1060 nanometer. We use any of these two. Either some manufacturers, some companies, they use 850, 1060. It doesn't matter. Both can image very efficiently the retina, the posterior part of the eye. So retina has different layers. Basically, retina has different classes of cells. These classes of cells appears as different layers. So you can see this is the OCT image of retina and the fovea. Fovea means where the light focus. Okay. So here is the visual sensation. Light falls here, and this region is the responsible for our vision. This is called the fovea. So you can see there are different layers here. All these layers represent different class of cells and their links. Okay. So, and at the bottom, there is a very important cell called, there are multiple cells called ganglion cell, bipolar cells. There will be a lot of artery. Artery means blood vessels supply blood to all these cells. And the most important part is called the photoreceptors. This is the most important, photoreceptors. So basically, we see something because our photoreceptors in our eye accept the light. So which cells is responsible for the vision? It is the photoreceptors. There are two types of cells. One is called the rod receptors and cone receptors. So corn receptors are very active cells, active photoreceptors or active cells, which is responsible for color vision. If you see something as colorful, you can see this is like a blue because the corn cells in your eye work properly. The rods is basically gives like dim light. Even if it's a little bit dark, it gives a very sensation. You can still see something on because the rod is activated with a very low light level. So there are two types of photoreceptor cells. So it is very important for the doctor to see this is functioning or not, and they want to see that everything is structure is intact and it is very well structured. That means it is a very healthy eye. So if somebody has a problem with the eye, first they go to the doctor. They don't immediately go to the OCT image. They first look into something called color fundus image. They will ask you to put your chin at some rest, and they will look into your eye. That is called the fundus photography. So they just look into that. They can see here. This is a very good eye. That means it has no disease. Why? Because it's like a very uniformly illuminated. It looks very healthy. You can see some structures. These are like a blood vessels. You can see a lot of blood vessels at the center. At the side, you can see a spot. This is called the optic disc. Optic disc means all the blood vessels are coming out of it. But the main region which is responsible for the vision is called the fovea, which comes here. Okay, so how a OCT looks like. So you take a OCT image across here and depth. So this is like a top image. This is called the top view. When you look into the eye, you can see only this. This does not include any of the depth information. To get the depth, what is inside this eye, you have to take the OCT. So once you have the OCT across this line, across the cross-sectional image, you can see like this. Here you can see this is the optic nerve, optic disc, and you can see the fovea like a dip here that I shown here. And all the layers are very clearly visible. There are multiple layers I will explain here. Okay, so there are photoreceptors layers are here. These are the posterior layers, and these are the inner layers, and these are the outer layers where the photoreceptors are coming here. So the doctor will look all this here. Okay, all the layers are clearly visible, and you can see different brightness. Different brightness depends on how much they reflect the light, scatter the light. So some layers reflect very strong light back to the OCT. I showed, right? The light comes and go back to the system again because the OCTs are basically a reflectometry based system. So the light go back. So some layers reflect on that appears as very bright. And you can see a lot of stuff here. These are the blood vessels. Okay. So this is only these scans, single these scans. That means a cross-sectional image. Okay. This is how it looks like in different diseases. For example, this is an 84-year-old person. He went to the hospital and he just told the doctor that I cannot see many things. Many places I can see like a dark spot. Some places I can see very beautiful image, but other places I cannot see like a dark spot. Then the doctor first did the fundus image color photography. Then you can see compare. Here it is very perfectly uniformly illuminated, very smooth, very clean. But here you can see some spots here, some weird stuff here. So the doctor advised the patient to take some kind of OCT image. So in the OCT, you can see here, see, there are some structure that's called undulations or the structure foldings you can see here. So these are like some growth in the eye. So there is like some growth here, some growth here. This is a very healthy one. But for this age-related macular degeneration, over the age, some deposit. These deposits basically come from the cholesterol. That's why the cholesterol is not only problem for the heart problem, it also affects eye. So the cholesterol will deposit in the eye and becomes like this. It also affects the vision. So this is the age-related macular degeneration that is over the age. This problem usually comes. This is called the drusen. Drusen is basically deposits of cholesterol lipids. Lipids. There are a lot of researchers, they really work what is in biochemistry of this one. Many people still really understand what basically this one. So somebody biochemistry who really want to do some PhD in future or some projects in the research, so they can still investigate what are these deposits. It's fine as a lipid, but there are some people still, they want to see what is, are they really lipids or something else there. So some people can also do in future, probably they can use these techniques or some ex vivo histology, some other imaging techniques to see what is inside. This also gives a very good result on that. And this is also retinal tumor. Okay, again, the retinal tumor is more common when you use the mobile phone a long time. This is a new study from US. Actually, if you apply the mobile phone or the laptop for a long time, like every day, early morning like that, it can also induce the tumor. Like, see, this is a tumor, cancerous. It can be cancerous or non-cancerous, but in this case, this is a cancerous basically. See, something has grown. So this is a fundus image. There is something here to more clearly visible in OCT image case. This is the way it looks like. and or retinal detachment. This is a problem. The people who do a lot of what is called physical exercise sometimes, especially at the older ages, when they get old, some people have, when they look at some picture, the picture will be shaking. So when the patient comes to the hospital, they ask like, hey, whenever I look something, the picture is shaking. Actually, it is not shaking, but when I look into that, the picture is shaking, because the reason is that then immediately they will take the fundus photography and show there are some dark spots here. I don't know, you can see in the computer screen, I mean, your screen. So when the doctor asks you to take some kind of OCT image, you can see the retina is detached. Retina is detached from its bottom surface. So there are other problems. And this is a diabetic macular edema. So this is for the diabetic patients. So if you have a lot of sugar, then your eye become like this. So it's very important. So this can only see with OCT images. There is no method, nobody replacing OCT to see this kind of structures in the eye. So this is really helpful to understand how the retina looks like and if it's affected by any disease or something. See, so this is very helpful for that one. Okay, now I want to, these are like I want to showcase some kind of OCT images for different diseases. And now this is a little bit on optics side and a new trend. This is like a new technology we developed and across the world on. This is called the adaptive optics OCT. Adaptive optics is a new technology, very new technology, super nice. It's not really, actually, it has been using for at least like 20 to 25 years, but people are still interested and we are still exploring what are the potential of using adaptive optics in OCT, not only in OCT, a lot of other applications. AO is, if somebody want to do some in future PhD or master project or something like that, this is one of the hot area they can do. If they're interested in optics, they can choose adaptive optics as a very great. I know some of the IITs, they also now started doing some research on adaptive optics. So adaptive optics has two applications. They used in two areas. One is the astronomy. The astronomy, because we used to image the satellite or interstellar, species or even planets, and remotely in the stars, we use the telescope. So the telescope, so we get the light from remote stars or remote planets from several millions of light years away or several light years away. And all this light is coming to us through the telescope. But once they reach here, the light will be very much aberrated. So when you look into that, the problem is that we cannot see it as a sharp image. Even if telescope can see it, we cannot see it like a sharp image. For example, if you look at the Saturn or Jupiter using your telescope, you can get a very cheap telescope this way. You can look into the Jupiter or the Saturn using the telescope. You can see them, but they are not very sharp because the light coming from them is coming through a lot of space, and there will be like a lot of turbulations coming on the way. And once it comes to the atmosphere has a lot of turbulence because the air is flowing, everything. So once the light comes to the telescope, it is called distorted or aberrated. So such an aberrated light comes when focused using the optics, using the the lens and all, the focus it. The problem is that we cannot see it's a sharp light. We can also experience what is meant by aberration. For example, if you look into something very long distance, for example, you can look at some headlight of a car or headlight of something very far away. You can see that light as not as a spot, but it's like a blur, right? Because our eye has some aberration. The light comes to our eye induces some aberration. We cannot see it as a very sharp. But if you look the same light using some kind of very small, very narrow hole, you can see it is like a very not as a sharp spot. At least like you can avoid that blurry like structure, like blurriness. You can remove the blurriness. You can see it relatively sharp, or you can still see it in a much better way. So this is basically called by the aberrations, because our eye has a lot of aberration. When light comes to our eye, it will get aberrated, and because of that, we cannot see many objects not as a sharp, but it is as a aberrated. So this problem is also in OCT imaging. So the first application of the adaptive optics is in the astronomy. It's widely used in the telescopes, world famous telescopes. And second application is the eye. Now it's also applicable for the other microscopic techniques. So why adaptive optics is important? So this is the eye imaging. So here you can see there is an eye. So we use the OCT for the light. So the OCT light comes here, and it focuses, because the eye has a lens and it focuses here. So we can get the image something like here using this sort of image. Okay. So in all cases, we want a very high resolution, lateral resolution images. So to get a very high light resolution, I told you before in the previous slide, the lateral resolution or the side resolution depends on a few things like numerical aperture, and it depends on the focus. Basically, this gives an equation that determines the spot size or the lateral resolution. So it's basically depending on the D. The F is the focal length of the light. We cannot do anything because the eye has a certain specific focal length, and D is the pupil entry diameter. That means it is the diameter of the light entering into our pupil size. We can also call it the pupil size. So to get a very high quality image, we had to use large diameter light coming into the light. Then only we can get a high quality image of the retina using OCT. So this is how it looks like. So when there is if we increase the size of the beam, OCT beam coming into the light, we should get very sharp spot actually. So if we increase the OCT light coming into the light, this is for imaging. Okay, when we image the light, if we increase the size of the beam, theoretically we should get a very small spot, very sharp spot. That means we get a very high lateral resolution. That means we can see the structures much clearer. But in effect, our eye induces some kind of aberrations. If you increases the size of the beam, entry beam, the problem is that instead of getting very sharp spot image, we are getting very blurry image. So this is a big problem in ocular imaging. When you image the eye with and if you want a very high lateral resolution, we have to use a very high diameter beam, large diameter beam, expecting a very sharp spot, but instead of we get a very blurry spot because this blurriness has come from basically the aberrations within the eye. So how do we correct it? So that method is called the adaptive optics. So we introduce a adaptive optics setup with the OCT. It corrects this corrects the aberration and it makes very sharp and how it looks like. So this is the adaptive optics actually. So you can see this is the light, reflected light coming from here. So any light reflected back from the eye has some sort of aberrations. I told that. So in adaptive optics, what we do is like we will measure this aberration. We are expected to get undistorted light or unaberrated light like this, very, very, very clean light actually. This is like a wavefront. These are called wavefronts. Okay, this is more on optic side. Okay, so when the light reflected, we expect very clean or unaberrated light, but in practice, what happens because of the aberrations of the eye, we get a very distorted light. So what we do is like this is the adaptive optics setup. So in this one, the adaptive optics has something called deformable mirror. Deformable mirror means its shape can be changed anyway. It's automatically, not automatically, using the computer, we change this is called deformable mirror. And it also using, this is called wavefront sensor. This is called wavefront sensor. There are different types. So this is more into the optics basically. This is called a wavefront sensor. So what we do is like, first we detect, first this aberrated wavefront will go here. And this is a beam splitter. Basically, this is a beam splitter. So when this aberrated wavefront comes from the eye, it first comes here and it goes here. Some proportion will be also here, camera. So we can see very poor quality image here. Now it's very sharp, but before aberrations, I will show you in the coming slide actually, how it looks like. So this aberrated light will be sensed by the wavefront sensor. This unit is called wavefront sensor. And this measure the wavefront sensor and give feedback to the deformable mirror. So based on this shape of the wavefront is reconstructed by the wavefront sensor, give us the feedback to the mirror. Then what happens, this deformable mirror will just conjugate to this wavefront. Conjugate means it makes the opposite of this wavefront deformation. So what happens, once it passes through it becomes straight. So basically we measure in adaptive optics, we measure the aberrations of the eye, the aberrations of the wavefront using a wavefront sensor and give us the feedback to the deformable mirror. So the deformable mirror will make the conjugate. Conjugate means opposite to this deformable, this deformation. So once it passes through this mirror, it becomes straight. So this is the way correction. This is called feedback. This is a closed loop. This is something we call the closed loop. So what eventually after this become very sharp. So such a sharp light, when we focus into the camera, when we collectively use the camera, we get a very sharp image. I will show you how it's done in OCT. So in the OCT, actually we have OCT light source, broadband light source. We have an interferometer, which splits into the reference arm and the other goes to the sample arm. So what happens to the sample arm? Once it go the light comes here, the reflected light will be very much aberrated, right? So when it comes, aberrated light will be collected by the adaptive optics unit. So how do they? The same process. So first, it will go to the wavefront sensor, the Hartmann-Schacht wavefront. So this is a very famous one. So this sensor will detect the wavefront deformation. Wavefront will be deformation. Wavefront deformation will be measured by the wavefront sensor, and it reconstructs using and it generates a shape of this wavefront basically with this deformation, and it gives us a feedback to the deformable mirror. So the deformable mirror will accordingly change its mirror. It is a bunch of mirror lights. That mirror light will change its shape. So eventually, when it passes through that, it becomes like a undistorted, very perfect wavefront. So such a perfect wavefront will interfere with our reference mirror, because our reference will not be undistorted definitely. So this will be interfere, and we get a very high quality interference fringes. And the way we process for the OCT image reconstruction, just process it and we can reconstruct. So this is some example. So if I'm not using any AO, adaptive optics, I'm getting an image like this. So this is I'm measuring from the photoreceptors. I just a couple of minutes ago I told like photoreceptors are the light cells, cones and rods that accept the light, and that's basically generate the visual signal to our brain, right? So without AO, we cannot see any cells here. But the moment you turn on the adaptive optics, you can see all the cells. So you can see like a small spot, white and dark spot. So these are the photoreceptors. Whatever you see as a white is the cones, and the dark as the rod photoreceptors. So you can see the difference. So how this improved? Because the lateral resolution of the beam has improved because of the adaptive optics correction. So there are several things I need to talk about here, but I'm just skipping all this, because if you are really interested, you can gently go to this one. You can browse it on the internet, and you get a lot of study materials and go step by step. And they are like mostly on optics. So you can clearly understand this is a very emerging and hot area. Many people are these days adopting it. So it will be really helpful for somebody who really want to do in optics, like some research in future. Okay. So this is another demonstration how the OCT. This is taken from my eye. How it's like this is AO off and on. How AO means adaptive optics. So adaptive optics also allows you to focus on. We can also focus the light on certain layers. So it has a lot of advantage basically. So here you see. So this is before and after. It is shaking because my head was shaking, because that's a problem with the human imaging. Sometimes we will not be steady. So sometimes there will be some shake, because this is coming from my head shake. My head is shaking, so it was not properly taken. So you cannot see any. You can see the different layers, but you can't see any kind of any cells here, because the lateral resolution is so poor. So I will show you what happens when the AO is turned on. Okay, so now you cannot see. Now you can see, because the moment you turn on the AO, you can see. Now this is the photoreceptors. So look at the photoreceptors. Now I'm focused the light on photoreceptor. You can see very sharp spots, because the lateral resolution is so much improved. The axial means the depth resolution is already stained. That's why you can see the different layers. It will not change, but the only thing, the lateral resolution is improved. See, now you can like the spot. This is for the photoreceptors. This is taken from my eye actually. So this is for the inner retinal layers. So you can focus here. This is outer retinal layers, that is photoreceptors. This is called the inner retinal layers. That means here you can see. Now I focus the light on here, and see how it's like. This is before. You cannot see any structures here, but now when you turn on the adaptive optics, you can see some cellular structures here. See, these are called the ganglion cells. It's very important. Ganglion cell. Ganglion cell is also monitoring. The doctors also these days monitoring the ganglion cell, because it's also affected by the glaucoma. So now the glaucoma has patients. This cells, glaucoma cells, will be destroyed, or it will be completely eliminated. So it has this also a biomarker. So now this days doctor also wants to see, okay, if there is any glaucoma, then they will immediately start looking into the OCT image and see there is a ganglion cell. So the ganglion cells are much more clear with the adaptive optics also. See how the adaptive optics is a kind of enhancement of the OCT images, and to see the more cells much more clear. The overall the image quality will not be, you see a lot of grainy like structure that is inherent to the OCT image, because OCT is an interferometry technique. You see a lot of grainy, like you can see a lot of noise, a lot of spots here. So these are basically called speckles. So if you use any coherent source or partially coherent light source, you can see something called a grainy like structure that is called speckles. So these are called the speckles. Okay. So there is a way to reduce this speckle and make this very sharp, and that's called despeckling. That's a different topic. I don't want to speak more on it. Okay. And this is just a final comparison of how the OCT looks like. This is just an OCT. This is just an OCT. You can still see all the layers of the retina, like photoreceptors, ganglion. There are a lot of cells. But these cells are much more clear here with AOCT. So this is with the AOCT. The top panel shows AOCT. You can see like these are like a stitch, because when you use OCT, your field of view will be limited. That's one of the disadvantages of the AOCT. Once you apply the AO, adaptive optics, you cannot take it for the whole retina. You can take only a small piece, then stitch together. So this is a AO image after stitching together. But when we use the AO, we can only take a very small region. That is one of the disadvantages, because the deformable mirror has a very limited number of pixels in it. That's a problem. So now the technology is advancing. Even with the there will be in future, there will be better AO wavefront sensor and also deformable mirror that we can image the entire retina in a single shot. That's okay. But you can see the difference. This is AO, this is OCT, and adaptive optics corrected. So we corrected the aberrations in the eye, optical aberrations in the eye using adaptive optics. Now the image quality is greatly enhanced. So these are something to show, okay, there are some improvements in the image, and now we can see the ganglion cells. This is very important to monitor the cells for the glaucoma. Okay, and this is another example. You see, the moment I turn on the adaptive optics, it corrects the aberration and it makes very, very sharp light. That means it improves the lateral resolution. You can see the cells much more clear, right? This is a photoreceptor cells. Okay, this is without, and the moment you turn on AO, it becomes very beautiful. Yeah, this is also from my eye. Okay, so and this is a final topic, and after this I will. This is a very emerging topic. In future, within next few years, we will hear about this technology. This is called optoretinography. You might never heard about it. This is a very emerging technique. We have started publishing a lot of great papers with this in science and nature. Like, because what is optoretinography? Optoretinography is a many of you may not sometimes understand this, but I just want to, since I mentioned the OCT eye imaging, I just want to mention this is a new area. At least it is good to hear about it. If somebody, like in future, they want to do some kind of great research or something, this will be very hot area. Especially this is useful for biologists, biochemists, chemistry people. This is a really great people. Even for the optics guys, you can do a lot of things. Okay, so this is a retina image and OCT image. You can see different layers are visible, right? So these layers, I just zoomed this area. I just magnified this area. You won't see it like a much better way, because the OCT, like you can see like this. But these are different layers corresponding to different. So the top reflection, the bright layer is nerve fiber layer. There is a lot of ganglion cell layer, inner plaxiform layer. There are different layers corresponding to different anatomy of the retina layers. See, these are different classes of cells here represent. And this area is the photoreceptor cell. Okay. So this is the photoreceptor. This is called a rod photoreceptor, and this is a cone photoreceptor. So the photoreceptors, photo means light, receptor means accepting. So the photoreceptor means these are the cells responsible for accepting the light into our eyes, and they generate some biochemical.
Long board, we have time. But in table tennis, the fraction of seconds we have to decide whether the board has to smash or whether the board has to
Wait, there's some sort of interruption. Is it to me?
Fraction seconds they will act. So the neuromuscular development will happen. Intellectual development will happen. So they have the same time the students are enjoying the game and their development is happening. That's why table tennis is the most important, most suitable game for the students, the beginning age. And the second one, which is not my opinion, the second one.
Excuse me. Can I talk now? Hello? I'm sorry, there is a... Oh, okay. It's came. Do you want me to stop now or what? Do you want to... Okay, thank you, thank you. I'm sorry for the interruption. It took a little bit lengthy for me. Yeah. Uh, I will soon finish it. Okay. Because it's interdisciplinary, I have to explain a couple of things much more detail. That's right. So these are the two photoreceptor cells, and this is a new technology to test whether, because the OCT can only show there are layers existing, but it cannot tell whether this is functioning or not. So this is a new technology or the new method called optoretinography. We are introduced the first time in place, actually, optoretinography, actually. So this is a method used to test whether these photoreceptors are functioning or not. How do we do it? So for that, we will shoot some light. We first dark adapt our eye. Dark adapt means we keep in the eye in the dark for some time. That means all our biochemical process in our eye will become like a very steady, and we will shine the light immediately. That's called stimuli. So during the stimuli, in response to the stimuli, what happens is that this is called the outer segment of the photoreceptor cell. It gets first compressed and it elongates. So this is a method to test whether this cell is functioning or not. So this is a very efficient way to test whether a photoreceptor is functioning or not, is to give a stimuli to the photoreceptor. So as a response to the stimuli, it first shrink and it expands. So by OCT allows to measure this displacement from the OCT images, and we can use it to confirm that the photoreceptors are functioning or not. Because this is very important for neural testing these days, and also for the stem cell therapy, stem cell research. So this is the way it works, actually. So how we test? So we expose some light to the cone after dark adaptation. In response to the cone, as I mentioned, the outer segment of this cell, photoreceptor, will expand. That we can measure using the OCT images, because the OCT also allows to measure the displacement. We can see it measure from the OCT images how much they displaced. So OCT basically gives us both intensity as well as a phase. So using the either intensity or phase, we can basically measure this displacement. So if there is any displacement, that means that photoreceptor cells are healthy. That means it is functioning. So these are for different bleach level. Bleach level means different light levels. So this is I just want to pass. So there is why this happened. There is a lot of biochemistry happening here, because in our corn there will be conopsin. Basically, they absorb the light and they undergo some kind of photoisomerism. There is a lot of biochemical process. The whole process is called phototransduction. So this is something very interesting. In future you can read about it. So because of this changes happening, and these changes can be monitored using OCT, and we can monitor this change in response to the light. So if we get a plot like this, that means that photoreceptor function is basically functioning. And we also these are some of the research I just want to show. This is optoretinography studies made in mice, actually. So this is another OCT system which does not make use of scanning. This is called full field OCT system. And using this OCT, I can show this. So these are the retina of a mice. So this is not human. This is a mouse, actually. So this mouse, using a OCT, we generate OCT images taken from this system developed. This is a very latest, one of the latest. You can show here. This is the outer retinal part. You can see when the light falls, what happens here. You can just look here. So when the light falls, see this band becomes very brighter, and these two bands slightly shift. So we can measure this shift related to some outer bands and plot like this. That means the photoreceptors of this mouse is functioning properly. Okay, so this is called the neural testing. This is a very new technique. It is called optoretinography. It is not even commercialized. It is not used in clinic, but it will be very soon, because we are also working with some companies. They also patent. We have some patents, and people are also working to adapt into. So in future, this is a method. So if you have, currently there is no way to know that the photoreceptors are functioning. Or there are some methods. It is called ERG, electroretinography, but it cannot tell. It can tell as a whole, but it cannot tell at a specific region the photoreceptor functionation or not. So this is a very important new method. It's a very state-of-art latest technology. to understand how the eye. So this is purely based on OCT technology. So the OCT can, using this technique, we can test whether the photoreceptor of the eye is functioning or not. Just repeating the same thing again and just telling that. Okay, so this is, I can show once again. If something happened like this, definitely the eye is functioning, eye is working or not. This is in mice. Okay, human is also, I shown in the previous slide. Is the curve looks like this. Yeah, so I just want to conclude. Like it's already took a long time, but I just want to show some of the areas in the research, interdisciplinary area with the optical coherence tomography. So optical coherence tomography is a very powerful non-invasive imaging modality that can be used for structural and functional imaging of biological tissues. And this property can be used for many clinical applications to diagnose, for like diagnostic applications, diagnostic imaging of diseases. And for OCT also helps to not only for detecting the diseases, it also helps to understand what are the different biochemical processes happening in the tissues. So this is very interesting for biochemistry, in the disciplinary research like biochemistry and chemistry people, or even biologists, those who are majoring at the biological sciences. And a lot of research is going on to explore the potential of OCT. Instrumentation, for example, adaptive optical OCT is a very great area, and its application. So these are some of the area I just want to share with you. I think at least you get a glance or some kind of flavor of OCT imaging and how it is used for different techniques. Although some application I took a little bit longer because it's a little bit interdisciplinary, so I just want to explain how it's like that. Thanks for listening. If you have any question, you can ask me now, or you can also text me later. Thank you so much. Okay. Also EE, I had to add like a triple. I didn't add one of my affiliation here, but it's okay. This includes a lot of optics. I didn't cover everything because every slide needs at least like 30 minutes to explain, but I just want to glance through the thing. There are something like this. There are several things interesting for the photonics people. That means optics, physics. Then electronics is now there is another progress I didn't mention, but now the AI is integrated, artificial intelligence is integrated with this one. Even without doctors, OCT can tell what is the potential eye disease you can have in future. So there is one big project is going on. That is there is a VR like devices, eye covering device, VR virtual reality like devices typically used for gaming and applications. So that itself is an OCT now. So they can automatically scan our eye and tell. So when you assess the eye, there is another advantage. Eye is a part of brain. The retina is a part of brain. So that's why many people are interested in retina research, eye research, because it's not because of the eye, it's because of the brain. Because retina is a part of brain. So any changes we see in the retina will be that also linked to the brain diseases. For example, Alzheimer's. So the Alzheimer's is memory loss over the time. Some people it will be prematurely lose the memories. That's Alzheimer's. So the Alzheimer's is basically caused by the deposition of there's a specific. There are a couple of chemicals actually deposits in the brain. So that deposit in the brain means that deposit we can see also in the retina. So just by looking into the retina, we can tell, oh, there are some problems coming in the brain. So this is the way that now these days people are looking the eye or the retina, monitoring more closely to the changes in the brain and also the cardiac rhythm. So all the cardiac rhythm can also monitor from the eye. So these are like a very emerging areas. Just to give, yeah. So feel free to ask me if you have any questions, and I'm open to any question. Yeah, please. I can hear. Yeah, I can hear you. Thank you. Yeah, yeah. Mm. Oh, yeah, dentistry. Dentistry. You mean, yeah, yeah, that's good. Oh, this is the one, dentistry. You are more interested in dentistry, right? Okay, the dentistry, because that depends on these applications use different wavelength band actually. Actually, I apologize, I didn't include that slide. Actually, there is one more slide that basically mention about different wavelength band used for OCT imaging. So, ophthalmology we use 850, 1300, and also 1060 nanometer. For the dentistry also we use higher bands, higher wavelength regime for like 1300, and also now 1700 also using. So if you use the higher wavelength band, that means if you go to the higher wavelength light can penetrate deeper into the tissues. That's the advantage. If you go to the lower wavelength, it can penetrate shallow. It cannot go much deeper. But in thick tissues, I think thick tissues like I mean the dental or the teeth or any other skin or something like that. So for a very hard look like tissues or even the samples, we have to use higher wavelength ranges. These are the images taken with 1300 nanometer. So the 1300 nanometer is most preferred for highly dense. And don't think that this is too deeper portion. This is like a very shallow. This is like only one, maybe like 500 micrometer or even 750 micrometer, not even 1 mm. So this depth means it's not really several millimeters. It will be maximum up to 1 millimeter or something like that. That's it. But it is still useful for many of the dentistry, dental issues to diagnose for that. Yeah, the light definitely goes. Any light can penetrate. It depends on the wavelength. So if your question is like, because we use NIR, we are not using visible light. These are NIR. NIR is like a higher wavelength band, which has the inherent properties to penetrate more into the tissues. That's a good point with the NIR we use. All the OCT use NIR. OCTs also, there are new OCTs also. A lot of people are working on visible light OCTs, but it doesn't have that much useful but use applications. But most of the biomedical applications use near infrared because it need a better penetration into the tissues. So NIR can penetrate. Mm. Oh, no, 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 no. Okay, okay. One more explanation. One more thing I need to do. Although I'm using optics, optic doesn't mean visible light. So when it's also a problem for many people. They misunderstand. When we use optics, means optic is not referred to the visible light or like anything we can see, but there are infrared radiation, near infrared, all belongs to optics. No, 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 no. No, no, no. That is visible range. That is visible. That is called the visible range. Visible, and we use near infrared. For example, 400. 400 is close to the UV. Actually, 400 to 700 is typically considered a visible range, and 700 to close to 1300 or 900 we call it as near infrared. Or 13 up to we consider up to 1400. It can call the near infrared. That's also considered as optics because we have optics for that. It is not really. Hmm. Okay, okay. Yes, it is tissue specific. It is tissue specific. Exactly. It is tissue specific because different organ has different tissues. So we look at the tissues then. So we don't look at okay, of course we look at the organ, but organ is made up of our organs are different organs are made up of different types of tissues. So it depends on the tissue. Some tissues are like very optically thick. That means very dense. Then we have to use 1300 nanometers. Some are not that much thick. Then we can use for lower wavelength. It's not a problem. Yes, absolutely. Absolutely. Yeah, yes. Not only absorption, there's a scattering, but mostly we look into the absorption. Yes. So there is I didn't show actually, you can also Google it and see like just absorption spectrum of visible. Uh, that's one of the reasons we don't use visible for all this one because the visible radiations like 400, 500, all this will be absorbed by different tissue components like collagen, water, hemoglobin. So our tissue has got a lot of components in it, chemical, organelles. So these have absorption in strong absorption visible So we cannot use it because whenever we shoot it, they will simply absorb it. So we cannot see, we won't get any signal back. That's the problem. Except some fluorescence. Some fluorescence we get it back. We have to use the visible light. But for the near infrared, we need to, our purpose is to light has to go back, reflect. So we use the NIR, near infrared. So once we go to the higher wavelength, it has better penetration because of the less scattering. That's one of the reasons. Once you go to the thick tissues, the major problem will be, of course, there will be absorption, but in addition to that, there will be scattering. Scattering will be a big problem. To avoid the scattering, scattering is inversely proportional to the wavelength. The scattering intensity is inversely proportional. There are two types of Rayleigh scattering and Meade scattering. So once you go to the Rayleigh scattering, it depends on the size of the particles in it, right? So if the Rayleigh scattering is inversely proportional to the force power of the wavelength. So if you go to very high wavelength, higher wavelength, large wavelength, what the will be less, right? So that means the light can penetrate deep into the sheath. So it is based on basic equations. Yeah, that's a really good question. And yeah, I mean, I didn't really include that because it takes even longer time to explain each and everything. I don't want to. I just want to give some just flavor of all these things. That's okay. But you asked a very good question. Thank you so much. Yeah. Yeah, feel free to ask if you have any questions. You can ask. No. Yeah, thanks for asking. This is basically only for imaging. This is only for imaging. Using this one, you can see that there are some problems, and you can also test whether that is functioning or not. For example, I showed you, right, the photoreceptors are working or not. We can do some kind of functional testing, and we can see the image, but it cannot be used for treatment. So what you ask is for the treatment. This is for the diagnostic imaging. That means we can see there is some problem just for imaging and functional testing. That's it. But we cannot use it for therapy or for the treatment. So for the cholesterol, yes, there is a new technique that is again laser based, but it is not OCT. OCT is for imaging. So we want to remove the blocks, right? Because we have the cholesterol deposit. Sometimes we will have it for older. Once somebody gets old, they have the calcium deposit. So our aim is to remove this. So there are two ways. One is the stent, right? Stem and balloon. There are different options. These days we don't use much that. We typically use the stent implants these days, employing. But even some cases we cannot put the stent because I told you the calcification has a problem. So if you put the stent, we cannot put that. That's a problem. So in that case, actually, now there is a new technique called high power lasers. So anyway, we are putting the light into it. So we use the same path for eliminating very high power laser. High means not very high, but it's like affordable. Affordable for the big blood vessels like artery. So using the high power laser, we can basically melt the calcium crystal. So the calcium crystals are like rock. And if you shine some very high focus light, high power lasers, pulse lasers, some companies are also building pulse lasers. So you focus it on, because of the huge power, high power, it's like, and you are focusing it, generate some heat and it can melt the calcification and it can be completely removed. I think I found it is very successful in some cases. I don't know if it's got FDA approval. Approval means for marketing and selling this product for the human trials. But I heard it's like some low power devices are already in the market. So using the lasers, you can remove, at least you can reduce the thickness of the blockage in the blood vessels after. Yeah, that's also a good question. Yeah, I really like it. No more question. Okay. Yeah, I mean, yeah.
Am I audible, sir?
Yeah, of course. Absolutely.
Yeah, my duty here is to deliver a lot of thanks. At first, I thank my esteemed resource person, Dr. Ritish K. Malapat, for sharing expertise on topic advances in optical coherence tomography and its application in modern physics. I think that will inspire us today. And I think main highlight of this session is he explained in such a simple way that everyone could understand. And he's very happy to explain about his studies and all. So thank you, sir. Thanks a lot for this session. And gratitude to Mohammed Shibu, conference chair and head of physics department for steering this event to excellence. And as I thank to all participants, students, teachers, and delegates who joined both online and offline, and your active involvement made this session a huge success. So thank you. Thanks a lot.
There is nothing here. You can put it on. I'm in a meeting. Yeah. Okay, no problem. Yeah, thank you very much, and thank you for giving me a chance to talk to you all. And it is a great opportunity for me to explain all this. I appreciate it. Thank you.